With this video, we're going to be looking at what is commonly called complex genetics. Now, in another video that I did, I talked about Mendelian genetics, which is also called simple genetics. And this is just giving you the idea of what it means to be homozygous dominant, heterozygous, homozygous recessive, what dominant meant, and what what recessive meant. Well, when we look at Mendelian genetics, this falls into what we call complete dominance. With complete dominance, the dominant trait is always expressed. Remember when we talk about genes, when something is expressed, that's the one that shows up. And so the one that always shows up is dominant, and the one that tends to hide is recessive. So when we look at complete dominance follows the rules of Mendelian genetics or simple genetics. So this is kind of what we've always talked about to this point. So I always use the example red times white is red. No matter what, red is dominant, hides the white. Red times white is red. And so with complete dominance, one trait is expressed over the other in a heterozygous condition. So in complete dominance, the dominant one completely hides the recessive trait. And so this is just like if you look at the videos on monohybrid Punnett squares, or like I said on my video that talks about Mendelian genetics or simple genetics, complete dominance is the example all teachers use when we're first teaching the concepts of genetics. But you guys know that the world is not red and white. There is some pink in there, or there's some red and white spots. So there are other types of genetic patterns, and this video is going to focus on two of them. So let's look at a complete dominant. So let's say we have heterozygous times heterozygous. So remember, heterozygous means you have a dominant and a recessive trait together. And let's say that big T is red and little t is white. So both of these plants have red flowers. So we would draw our Punnett square. Remember, 2 times 2 is 4. That's how I always remember. And you fill it in. So in the end, this is the results of our Punnett square. So remember, we can look at the genotypic ratio. Remember, genotype is where you look at what type of gene you have. So remember, in this case, our GR is 1 to 2 to 1, because I have one chance of having a plant that's big T, two chances of having a plant that's big T, little t, and one chance of having a plant that's little t. Now that's different from the phenotypic ratio. Remember, phenotype is what the plant actually looks like. And so in this case, red flowers versus white flowers. And since red is dominant, my PR is 3 to 1. I have 3 chances of having a plant with red flowers, because 3 of these boxes has a big T in it. And I have 1 chance of having a plant with white flowers. Okay? So in complete dominance, the dominant one always wins. Now, that's different from what's called incomplete dominance. Okay? With complete dominance, the dominant trait completely hides the recessive trait. In incomplete dominance, it doesn't. So complete dominance, red times white is red. In incomplete dominance, red times white equals pink. So with incomplete dominance, the dominant allele, the dominant trait, does not completely hide the recessive one. So you get kind of a mixture. You get kind of a blend of the two. So the Punnett squares are still going to look the same, but you have to know what the question is asking. So for example, we're going to use the same one we just did. Okay. Our Punnett square is going to look pretty much exactly the same as what we did in the previous example, but this time my answers are going to be different. And what I mean by that, okay. This Punnett square looks just like the one we just did. When we talked about complete dominance, my GR was 1 to 2 to 1, and my PR was 3 to 1. Well, this is now incomplete dominance. So in this example, big T, little t is pink. Okay. In incomplete dominance, big T is red, big T is pink, and little t is white. Okay. So my GR, my genotypic ratio, is still 1 to 2 to 1, because I have one chance of getting a plant that's big T. I have two chances of getting a plant that's big T, and I have one chance of getting a plant that's little t. However, now my PR is going to be different. Okay. My PR is now also 1 to 2 to 1, because in incomplete dominance, I have one chance of getting a plant that's red, I have two chances of getting a plant that's pink, and I have one chance of getting a plant that's white. So when you're doing genetic problems, make sure you read what the question is asking. If it talks about a red plant and a white plant and their offspring being all red, that's probably complete dominance. Red times white equals red. Or if it talks about, in the question, it says incomplete dominance, or it says like black and white and you get gray, or brown and white and you get tan. Okay. Well, if you have brown and white crayons, you get tan. If you have black and white crayons, you get gray. So those are combinations, those are mixtures. That would be incomplete dominance. So you're going to do the Punnett square for both of them the exact same way, but you have to watch what your answers end up being. If this was complete dominance, our phenotype would be 3 to 1, 3 reds and 1 white. But this is incomplete dominance, so our phenotype is 1 to 2 to 1, so 1 red, 2 pinks, and 1 white. Now there's still a third example as well. Now with this Punnett square, we do have to draw it differently. This is what's called codominance and multiple alleles. Okay. When we teach you genetics, we tend to just use 2 alleles, like a big T and a little t, a dominant and a recessive. However, you know that nature is this wonderful mix. We have more choices to choose from. Okay. There's lots of different flavors of ice cream out there. Okay. Complete dominance would be like just having chocolate ice cream and vanilla ice cream. We have a dominant trait, which is chocolate. We have a recessive trait that's vanilla. But you know there's lots of different ice cream flavors out there. That's because people want variety. Well, nature wants variety as well. So in all honesty, there are more alleles to choose from out there than just a big T and a little t. And this is what we call multiple alleles. Okay. So remember, nature likes variation. Nature doesn't want everything to be just chocolate or vanilla. Okay. Nature wants to have strawberry, nature wants to have marble, nature wants to have rocky road. Nature likes variation. Okay. So we really have more than just 2 alleles per gene. Okay. So a good example of that would be blood typing, which is what we're going to show here in just a minute. Okay. You know that you can have A blood, you can have B blood, you can have O blood, you can have AB blood. Oh, AB, that's 2 dominants together. Okay. Nature gives us more than just 2 choices. So this is what we call multiple alleles. Okay. Remember, if you're not familiar with the term allele, think of an allele as half a gene. Genes are recipes that tell you how to make proteins. And we've all seen recipes before. The recipes include the instructions, and the recipes include the ingredients. And you have to have both parts of the recipe in order to make the recipe. You get half the recipe from your mom, you get half the recipe from your dad. So think of an allele as half a gene. You get 1 allele from your mom, you get 1 allele from your dad. And so these letters that we're using represent alleles. So if you're big T, you have both parts to the recipe. Big T, you have both parts to the recipe. Well, there's more to nature than just having 2 letters to choose from. Think about all the different eye colors we have, all the different skin colors, all the different hair colors. Okay. Now, those aren't multiple alleles, but this gives you a good idea of what we talk about when we talk about how nature likes things to be different. So nature actually gives us more than just 2 choices when it comes to the ice cream. And so this is what we call multiple alleles. And like I mentioned, blood type is the major example that we use to talk about multiple alleles. Okay. With blood type, you could have an A gene, a B gene, or a 0 gene. Now, what this is actually talking about, when we look at blood cells, all cells have things called antigens on their surface. And those antigens are proteins. And proteins are used to let nature know, okay, what kind of cell is this? So when nature sees a cell that looks like this, it goes, oh, you're a blood cell. So everybody on Earth has the same proteins on their blood cells. That's how nature knows it's a blood cell. Well, we've known about blood transfusions for years. There's actually hieroglyphics in ancient Egyptian pyramids that we think represent blood transfusions. And sometimes blood transfusions worked, and sometimes blood transfusions don't. And so a gentleman named Carl Landsteiner is the gentleman who is credited with discovering that we actually have 4 different genes for blood type. Well, like I mentioned, to be a red blood cell, you would have a protein on you. And so let's say you had these half-moon proteins. These are called antigens. So when nature sees this, it goes, oh, you are a blood cell. But then he discovered that some blood cells had an extra protein on it. Well, he discovered that protein first. Think of it as an A. Well, other people had a different shaped protein on them. Okay. Let's take this one and turn it into something that looks like this. Well, he discovered that one second. He called that B. While some people didn't have any extra protein on them, so 0 extra proteins on them, and that's what we now call O. So you could have the A gene, you could have the B gene, or you could have no extra gene on you at all. So these are your multiple alleles. So we have 3 choices, we have 3 alleles. Well, nature has 2 dominant alleles, the A and B, and 1 recessive, the O or the 0, to choose from. It is possible for 2 dominant alleles to come together in 1 gene. And this is what gives us our AB blood type, and this is what's called codominance. So people with AB blood have both of the 2 special lipid/sugars, proteins, on the surface of their red blood cell. And both traits will be expressed equally. So now red times white equals red and white spots. So this Punnett square is going to look a little bit different. Okay. If you have A blood, you could be big A, or you could be big A0. Notice multiple alleles. There is no little a in codominance. If you have B blood, you're big B, or big B0. If you have O blood, the only thing you can be is 0. And you have AB, you are A and B. So we have 3 different alleles to use here. Okay. Notice none of them are little. So therefore, our Punnett square is going to look very different. So when we do a codominant Punnett square, let's say we have a female who has A blood, and we have a male that has B blood. Well, we just can't do a Punnett square with just based on that. We have to know more about her. So let's say she's homozygous, which means she is big A. But he's heterozygous, which means he's B0. So we're going to draw our Punnett square like normal, but it is going to end up a little bit different. Okay? Now, again, when you do your Punnett square, it doesn't matter which one goes on the top, which one goes on the side. I usually put the first one on the top, and I put the second one on the side. Now, I always draw a line through there to represent a 0 because it has 0 extra glycoproteins on it. So if this was these two people, they could have a child with AB blood type, and they could have a child with A blood type. Now my genotypic ratio would be 2 to 2 because I have 2 chances of having a child that's AB, and I have 2 chances of having a child that's A0. My phenotypic ratio would also be 2 to 2 because I have 2 chances of having a child with AB blood, and I have 2 chances of having a child with A blood. So in this case, they have no chance of having a child that has B blood like the father in our example. Now, another thing we could do, let's say we have a mother who has AB blood, and we have a father that has O blood. We would draw our Punnett square, AB, 0, 0, A0, A0, B0, B0. So again, our genotypic ratio would be 2 to 2, 2 chances of having an A0 and 2 chances of having a B0. Our genotypic ratio would also be 2 to 2, 2 chances of having a child with A blood and 2 chances of having a child with B blood. So there's lots of different examples you could do with this. But remember, with incomplete dominant, you are going to have a recessive letter, big T, little t. But with codominance, you're going to be using 2 different letters. So make sure you're watching the examples or what the question is asking so you'll know how to draw your Punnett squares. So just some examples of codominance. Remember, complete dominance, red times white is red. Incomplete dominance, red times white is pink. Notice in codominance, you're going to end up with both showing up. Co, meaning both. They're both going to show up. So here we have a red and white flower. It's not pink, it's red and white. Here we have a cow that's brown and white. Not tan, but brown and white. And like I mentioned, with blood typing is also codominant. So it's all based on the extra little proteins and sugars that show up on the surface of the cell.